Hello, welcome back. We are going to continue with our discussion of Chapter 11, taking a quick survey at Archaea and some of the groupings that it has in different classifications. So first of all, remember Archaea are a type of prokaryote, so no nuclear-bound, membrane-bound nucleus or other organelles. So structurally similar to bacteria, but they do have some differences. Remember, you can go back and take a look at the end of Chapter 3 as we went over the differences between a bacteria and Archaea. So just to review, let's see, lack of peptidoglycan in their cell walls. They have branch hydrocarbon chains in their membranes, and their amino acid methionine is an N-formal methionine. No, bacteria use an N-formal methionine for that AUG codon, whereas Archaea just use regular methionine. So when we take a look at these, I'm just going to pick a few groupings. Know that most Archaea are not extremophiles. We're going to take a look at what's called extremophiles. They just live in regular environments. Some of them are pleomorphic. They don't really have a set shape. We know they don't contain peptidoglycan. Looking at my notes here. But I'm going to take a look at 3 different extremophiles because they're kind of the most interesting ones. What's exciting about a boring old Archaea that just lives in the dirt? So no, the majority of Archaea are those boring old ones that probably live in the dirt, but there are some pretty cool exceptions to that. So we're going to take a look at those. The first group are called thermophiles, thermophiles and hyperthermophiles. So thermo means temperature and phile means to love. So like a hydrophilic loves to be around water. So a thermophile and hyperthermophiles love really hot temperatures. I have some numbers here. So they require temperatures over 45 degrees Celsius, which is about 113 degrees Fahrenheit, if you don't know your Celsius degrees, which I'm not super good with either. So over 45, which is really, really hot. That's hotter than most, bath household waters should be. There are the hyperthermophiles require over 80 degrees Celsius, which is 176 degrees Fahrenheit. And then there are some that can spend upwards of 2 hours at 130 degrees Celsius, which is 266 degrees Fahrenheit. That's the record holder for about 2 hours before it can start actually getting some of its enzymes denatured. Because if you remember enzyme function from your general bio, high temperature was one of those things that denatures enzymes. So how the heck is it that these organisms can spend their whole lives living at degrees over 100 degrees Fahrenheit? It's pretty amazing. Well, they have adapted the ability to maintain those covalent bonds and hydrogen bonds and interactions to, that's their optimal temperature, is to be really hot. I wore my Yellowstone shirt today because a lot of the organisms that you see in the Yellowstone thermal pools are thermophiles, and they are what are responsible for the beautiful colors of those different hot springs. If you've ever been able to go to Yellowstone, you know what I'm talking about. So those are the thermophiles and the hyperthermophiles. Okay. The next group of extremophiles are called halophiles. So halo, not angels, salt. So these are salt lovers. So these are organisms that can live in really, really salty conditions. Now, again, the reason why this is extreme is because most organisms would die in a hypertonic environment, right? So you remember our osmosis conversation, your prior knowledge. If you put a kind of freshwater organism in a hypertonic solution, water is going to leave out of that cell to try to create an equilibrium with the environment. The cell is going to shrivel and it's probably going to die. Halophiles, for them to be able to live in these really salty environments, their cellular parts, their cytoplasm, has to be just as salty as their environment. They have to be isotonic. So if they're living in like 20% sodium chloride, like in these salt evaporative pools to harvest salt, that means their cytoplasm has to be around 20% sodium chloride. Otherwise, they lose their water too quickly to the environment. So halophiles love salty environments. And there we go. Our concentration, our sodium chloride is about 0.9% sodium chloride. A lot of halophiles require, what do we see? Greater than 9%, so 10 times saltier than our blood and our tissue fluids to be considered a halophile. And there are some that can live all the way up to 17 to 23%, sometimes even 30% salt concentration. So they have really, really salty cytoplasm to stay isotonic with their environment. All right, and our last group of archaea that I want to talk about are called methanogens. And these aren't necessarily extreme environments, and not extreme physical environments, but extreme kind of chemical environments, in that they have the ability to produce methane. They break down organic material, and methane is a byproduct. So methanogens, methane generators. So I have found a couple of examples to show you where we might find these things. So here's one. If you take a look at its name, it might be, it's a really good descriptive name. So the genus is Methanothermobacter, thermoautotrophicus. Put that on your Scrabble board. So methano means it's a methane producer. Thermobacter probably means it's a bacteria that loves to be in hot situation. So not only is it methanogen, but it's a thermophile. Thermo, again, lets us know that it's a thermophile. Autotrophicus, autotroph means it can generate its own food. So like photosynthesis, but probably with chemicals instead of the sunlight. And if does anybody know what this is, right? So if you've ever taken a tour out at our waste treatment plant, this is a waste treatment plant. It's a kind of a sewage processor. So methanogens have the ability to break down organic material like human waste and produce methane as a byproduct. This other organism over here, Methanobacterium, so that lets you know it's a methane producer. Ruminatium, ruminants are a type of animal that they kind of chew their cud and they swallow and then they ruminate on it and then they chew it like cows and camels and sheep and stuff. So they live in the intestinal tract of these ruminants. So Methanobacterium ruminatium. And if you'll notice what this little cow is doing, he's farting. Cows fart, sheep fart, most mammals fart because they have methanobacters. They have these methanogens living in their intestines that help to break down organic material, producing methane as a byproduct. And it actually is a greenhouse gas. So animal flatulence is a big contributor to climate change and global warming and increasing the amount of these greenhouse gases in our atmosphere. All right, so those are our three groups of archaea. So the thermophiles, the halophiles, and the methanogens. All right, so we're going to come back and take a look at different groups of bacteria to wrap up our chapter 11. Okay, bye.